Well, thank you so much. You have no idea how glad I am and how blessed that my mom got to see and hear all this. But so the importance of clean water to global health and to economic development cannot be overstated. And while there is a myriad of contaminants that can make you sick, by far, perhaps on a global basis, the most important are pathogenic microorganisms like virus, bacteria, or pathogens. For about 1/2 of all the hospital beds in the world are occupied by someone who got sick by a waterborne disease. So today I want to present to you a vision about how nanotechnology can help us address this growing challenge, and also pitch for the importance of a fundamental understanding between bacteria and nanomaterials, not only so that we can come up with more efficient approaches for chemical-free, greener disinfection that doesn't cause harmful disinfection by products, or to enable corrosion and biofouling control methods, but also so that we can enable a proactive risk assessment to minimize the unintended impacts of incidental or accidental releases of nanotechnology. So in other words, I want to talk to you about the convergence between nanotechnology and microbiology within a context of environmental applications and implications. So let me begin by reminding you that perhaps one of the most important contributions of engineering of the last century was drinking water treatment and sanitation. That itself increased the expectancy of Americans from about 47 years old to, I think we're at 78 today. So you guys, we are working on the correct side of the decimal point. But unfortunately, we have not won this war. Clean water is becoming harder to come by due to a wide variety of factors. For example, population growth with a growing demand, climate change, aging infrastructure, and a wide variety of emerging pollutants. And this underscores the need for novel water management approaches as well as technological innovation. And this is where nanotechnology comes in. The word itself has a Greek origin. Nano means dwarf, or that's why in Spanish we say enano. But mathematically, we take it to mean 10 to the minus 9. In other words, we're talking about very small scales here. The National Nanotechnology Initiative defines nanotechnology as the understanding or control of organic matter, working atom by atom to create novel structures that have novel properties and enable novel applications. So for example, if this is a human hair, this right here is a nanofiber that can be used to transmit energy or information, or perhaps to reinforce concrete so it doesn't crack. And the thing here is that at this scale, you have some unique size-dependent properties. So materials that are inert, like gold, become catalytic. Materials that are insulators, like carbon, become conductors. Materials that are opaque, like titanium dioxide, become transparent. And our vision is to take advantage of these unique size-dependent properties of nanomaterials, such as superb sorption properties, hypercatalytic abilities, and so on, to create high-performance water treatment and reuse systems that enable a distributed differential treatment paradigm in a way that requires less infrastructure, less energy, and less materials. So in this axis, what I'm trying to show in this domain is that in the x-axis, we're showing how much you're spending for water and water infrastructure. And here is how advanced the nanotechnologies are. And what I want to point out is that nanotechnology can help both developed rich countries as well as developing countries, even though we recognize that they have different priorities. In the developing world, or in rural areas, people worry primarily about disinfection. And in, of course, Orange County, we worry about emerging pollutants, and a lot of, we use very high advanced technologies. But in both cases, nanotechnology can help us because both face the need to treat the water at lower cost and do better performance. In other words, to move left and up. So for example, in developed countries, it can help us modernize the aging infrastructure, make better membranes that have self-cleaning properties, that have multifunctional purposes to get rid of recalcitrant emerging pollutants. And in the developing world, we can, for example, develop point-of-use devices that take advantage of solar energy for disinfection, or perhaps, let's say, use magnetite to remove arsenic from groundwater. So these are some of the examples that we have been developing at RISE. A while ago, we test piloted in Guanajuato, Mexico, this sand filter that was coated by nanomagnetite to remove arsenic, and it worked pretty well, except for the fact that in some wells there was a lot of silica, and silica absorbs and competitively inhibits the absorption of arsenic. But it was, by all means, a successful pilot study. With chillingly, we have been developing fouling-resistant membranes by loading them with, among other strategies, antimicrobial nanoparticles. Of course, the driver here is to filter the water with less energy and increase the economic life of the membranes. With Mike Wong, we have been developing this hypercatalysts. He has been taking the lead here, which are 100 times more efficient than any palladium catalyst, and they are resistant to fouling. We're testing it at the hazardous waste site by DuPont. And what I'm going to tell you about it, since I'm at RISE, I got to tell you about Fullerenes a little. These are these little bucky balls that look like a soccer ball. They have 60 carbons. And basically, they're very good photocatalysts. And by that, what I mean is that they can take light, including sunlight, to generate reactive oxygen species, like singlet oxygen in this case, which are very effective at killing bacteria or virus. This was discovered at RISE. This earned the Nobel Prize to Rick Smalley and Bob Curl and Sir Harry Crotto. And what we did is, using the Bingo reaction, we added a functional group that makes it water-soluble, accessible to reactions in water, and it has a positive charge to attract to the battle zone, if you wish, the negatively charged bacteria and virus. So this is very effective, even with sunlight, to degrade virus. Now, of course, when you, and what I like about this is that you can use it in off-grid applications in remote areas where you don't need electricity, which is where you need the most, disinfection. Of course, there are two potential barriers that you need to think about it if you use this for drinking water treatment. One is the cost, and two is what if this is released and you end up drinking it? Well, we have done a lot of tests, toxicological tests, and we know that in the dark this is harmless. But nonetheless, you can address both of those concerns by immobilizing the nanomaterials. Remember, regarding cost, it doesn't matter how much a gram of this costs. What matters is how much it costs you to treat 1,000 liters of water. So if it's immobilized and you can reuse it and reuse it and reuse it several times, then the cost of treating water can be reasonable. And we were successful at immobilizing it here through a covalent binding. And now we're testing it in Swaziland. This is to enable safer peri-urban irrigation. This is a reactor that is a fluidized bed packed with photocatalysts. This is serving basically a small agricultural plot of 2 hectares for about 16 families. But one nice surprise that I thought might be of interest to some utilities here is that these photocatalysts are very effective at degrading pharmaceuticals and endocrine disruptors that break through wastewater treatment plants, particularly those with phenolic groups at high pH. The singlet oxygen is a very selective oxidant. And as you can see, that's a pretty good job as a polishing approach. So this is something, our philosophy is to try to innovate in ways that we can use the existing infrastructure as much as possible. And your wastewater treatment plants already have UV disinfection systems. So basically modifying and retrofitting it will be very easy. We also can use it as a pre-treatment approach. We got interested, of course, being in Houston by the recent oil spill by BP, the accident in the Macondo well. And as you know, there was a lot of weather oil that was left that was very, very recalcitrant to degradation. And usually the rate-limiting step for degradation is the hydroxylation of these hydrocarbons, the first step in degradation. So we thought that maybe if we use a photocatalyst to do that first step, hydroxylate the hydrocarbon, what that does is that it makes it more soluble, more bioavailable, and therefore more biodegradable. And therefore, we exposed the weather oil from Barataria Bay and saw that indeed the solubility was increasing after only 1 day of contact time, and the extent of biodegradation measured here by respirometry also increased significantly. So this also looks promising. Now, switching gears very quickly, we are at a unique, probably, time in terms of responsibilities of opportunities to steward nanotechnology as a tool for sustainability and prevent it from becoming an environmental liability, and that requires taking a proactive risk assessment. Because history is really full of so many promising technologies and policies that created a lot of collateral damage to the environment. And here are two of my favorite examples. Paul Hermann Mueller won the Nobel Prize in 1946 for using DDT to fight malaria. And you know what DDT did. And then we got Thomas Migley. I'm sure many of you know who he is. He was a mechanical engineer who was an amateur chemist, and he worked for General Motors. First, he added lead to gasoline as an anti-knock agent, and then he invented Freon, a chlorofluorocarbon. So many historians believe that no single organism ever did more harm to the atmosphere than Thomas Migley. But anyways, this is important to take this proactive approach, and we have been doing this by considering how nanomaterials interact with bacteria. And this is important because bacteria are the basis of all known ecosystems. They provide ecosystem services worth trillions of dollars, ranging from primary productivity to the biogeocycling of nutrients to degradation of waste and so on. And they also happen to be very convenient models to study the mode of action of how microorganisms might harm a cell. I want to give you an example by considering how bacteria interact with the most commonly available commercially nanoparticle, which is silver. Silver, you may know, was the antibiotic of choice before penicillin was invented. In fact, our pilgrims used to drop a silver coin on their milk to prevent it spoiling, to make it last a little longer. And many Europeans had all these legends, the only way you kill a werewolf is with a silver bullet. So some people are using silver, for example, in textiles to prevent bacterial activity, for example, socks from smelling bad. I think that's sort of a silly, perhaps, application. There are applications that make more sense. They add silver to baby drops when you want to disinfect without strong oxidants. And we have used it successfully in water filtration membranes to prevent biofouling. But what I want to talk to you for the scientists in the audience, just a very brief couple of slides only, I want to address one of the most elusive questions that we have faced in nanotoxicology, which is the antibacterial mechanism due to the particles, the nanoparticles are, or is it due to the ions that are dissolved? Very briefly here, the silver itself gets oxidized in the presence of oxygen, and then silver ions are released. And these silver ions are known to have a high affinity for thiol groups in proteins and inactivated proteins. That's how they kill the microbes. And the question is, this is toxic, but how important is the toxicity of the released silver ions that dissolves versus the nanoparticle? But the one thing I know is that silver itself, zero-valent silver, has a solubility of 0. So if you can make nanoparticles under anaerobic conditions and test them under anaerobic conditions, there should be no release of silver ions, allowing you to separate the effects, kind of take a reductionist approach, if you wish. And by doing so, we realized that the silver nanoparticles that were prepared anaerobically were not toxic at all to bacteria. Of course, if you took them outside of the anaerobic chamber and exposed them to air, then they became toxic, and the longer they were outside, the more toxic they were. So what that tells us is that the key determinant of toxicity is the ion and not the nanoparticle. And basically what that tells you is that you can basically test different kinds of nanoparticles of different sizes with different coatings, and you can explain the dose response by looking at the amount of silver released. So the practical implication is that you could engineer particles to release just the right amount of silver so that you don't essentially ends up wasting silver and also creating an unintended environmental impact. But also it's important to recognize from a Nietzscheesque perspective, if I may say, that whereas you don't want to overshoot or add too much silver, which will be wasteful and detrimental to the environment, you don't want to add too little either. Because there is a point at which you actually enhance the survival, you create resistance in the organism. We believe that this is probably due to poorly understood phenomena known as hormesis, which is the basis of some Oriental medicinal practices, where they feed you a poison at very low concentrations to induce a repair mechanism that repairs not only the damage done by the poison, but other unrelated damage. So you end up with a more fit cell. So you don't want to overshoot. So anyways, getting back to nanotechnology-enabled water treatment, NEWT. kind of like Newton, we're going to have to follow a tortoise path, like any technological innovation, where we're probably going to face a peak of inflated expectations, and we might be here until you get to that oops moment. And perhaps after that, if you stick with it, you get to a plateau of productivity. But the main barrier that we're probably going to face, that I want to touch briefly for the consultants here, is the cost. And related to cost, the cost of nanomaterials have not really decreased over the past years. For Fullerenes, it did for a while. There was a new technology, a furnace-based technology that brought the cost down, but then they went up. And what is important to recognize is that the cost of making nanomaterials is very, very cheap. What is expensive is separating them and purifying them for research purposes. That's pretty much the only way you're going to get to publish if you're working with a very purified nanomaterial. But we need to understand what are the trade-offs between purity and performance. There's clearly is some diminishing returns associated with ultra purity here in regards to performance. So for example, I can make these fullerenes, amino fullerenes that I showed you earlier, using very pure starting materials, or I can use soot of fullerenes, which cost 20 times less, and make it for 5% of the cost without sacrificing much the performance. So we got two ways of fixing this. First is being able to reuse and recycle, and the other one is do not get caught on the diminishing returns of ultra-high purity. So to conclude the technical aspects, I would like to say that the convergence of nanotechnology and microbiology does offer a great potential for meaningful disruptive innovation, which is important. And for example, we can come up with greener disinfection approaches that do not create harmful disinfection products, advanced photooxidation processes that destroy highly recalcitrant emerging pollutants. We can make nano-patterning of surfaces to emulate the shark skin or the skin of the lotus leaf to make surfaces that are resistant to corrosion or to microbial fouling. We can make membranes for filtering water that have self-cleaning properties, and perhaps in the future materials that sense the pollutant and attack it selectively on demand. Now, so overall, this, I think, is very important to enhance the competitiveness of our industry, of our entrepreneurs in the emerging markets of global health, of water infrastructure modernization, and of biocorrosion and biofouling control. On a personal basis, I am deeply honored and humbled to be the recipient of the Clarke Prize. This prize for me is really an inspiration for generosity, for making the world a better place, for making access to more people cleaner and affordable water. So for that, I am very grateful to the late Natalie Richardson Irvine Clarke, and of course her daughter Joanne, and James and the whole family, as well as the National Water Research Institute for making this award possible. Very, very grateful to the Institute staff. They were just superb, Dondra, Gina, and of course, everybody was so professional and made this a very, very positive experience. And of course, very, very grateful to Manny Elimelech for nominating me for this great honor, and for the committee and the people that wrote letters. I'm eternally grateful for that. I was very lucky to have been educated at great places. As a child, we used to move from one country to another, Argentina, and so my father worked for Shell Oil. And I was fortunate to have been educated by Jesuits all the way through, who instilled in me early on the importance of world affirmation, the commitment that the world can be a better place and you can do something about it. I got a great start in engineering at McGill University in terms of fundamental principles, but I wouldn't be where I am today without the education I got from the University of Michigan. I learned that commitment to excellence from Walter Weber. And of course, I was very inspired and learned a lot about principles of microbiology from my advisor, Tim Vogel, who sends his regards from France. But I was extremely lucky to have been mentored at Iowa. I mean, we had there Jerry Schnorr, his wife Jana, where are you? There you are. And of course, Gene Parkin and Rich Valentine. They told me about rigor and relevance and relaxation, RQ. And a testament to their success is the fact, I don't know if you know this, but the last 3 Clarke Prize winners have had very strong connections to Iowa. I don't think it was the water in Singapore, Jerry. Now, I am very, very grateful also to my friends and colleagues at Rice University to introduce me to the wonderful world of environmental nanotechnology. And I'm going to include here Mark Wisner, who is now at Duke, but he's still part of the family. He essentially was there when it all started. And, of course, all of the work, I did it all by myself without any help from anybody. Not. No, I do have to thank my great students. They are doing very well. And postdoc, for example, I'm very proud of Shaylee, who is now an extremely successful assistant professor at UCLA, and many others that came before and after her. But above all, I want to thank my family for their unconditional love, patience, and contagious happiness. And I am so lucky and so blessed that they're all here. I am very grateful that you're here, and I thank you for your attention.